Hello, in this video we will discuss about the vitamin physiology, vitamin A physiology. So what is the vitamin A? Basically the vitamin A is the fat soluble vitamin, the lipid soluble vitamin, which that is just like a vitamin E, as well as vitamin K and vitamin on the other hand vitamin D, which that is all vitamin is the fat soluble or lipid soluble. And here the vitamin A type is the retinol, and there are different type family of the vitamin A: retinal or retinaldehyde, because the aldehyde group are attached, and retinol contain alcohol group attachment, and the beta carotene, which that is contain two ring opposite, and there are many hydrocarbons, so you can see and understand. On the other hand, the retinoic acid are also present because the carboxylic group are present in the end of the hydrocarbon chain. And here you can see the methyl group attached with the ring, and double bond and single bond alternatively. So in this way, this is the type of the vitamin A with the different group will be attached to show the different their function. But let's begin to understand collectively. So here is the vitamin are an essential non caloric or organic nutrients essential in minimum amounts in the diet, and were discovered at the beginning of 20th century. So these vitamin A are present in the different food, for example, carrot, which that is in the form of carotene, and the milk also present, and egg yolk. So this all food is contain vitamin A, and vitamin A is essential for our body, for visualization, vision, for eyes, and on the other hand, the antioxidant. So here is I am drawing the digestive tract, which contain pancreas, duodenum, and this is the esophagus, stomach, and this is the duodenum. Duodenum is used for the absorption of vitamin A with the lipid. So here is the gallbladder and bile duct. So this is when enter the food into the stomach after the chewing, after the mastication, to become a bolus. This bolus contain a partially digested food. Carbohydrate partially digest through amylase enzyme, salivary amylase, and further go into the duodenum after the protein digestion to become a chyme. And this chyme will be further digest the fat through a lipase enzyme from the pancreas, and on the other hand the other proteases and the bile pigment, which that is used for the emulsification of fat to become a chylomicron, a chylomicron. This chylomicron will move into the lymphatic system first, and this is the lipid droplet will enter after the digestion into the lymphatic system and then go into the blood circulatory system. Why? Because the infection, and there are many refinery systems through our immune system, so the lymphatic system is preference. So here is the carotene, which that is go into the digestive tract, and this carotene will enter through a CD36 and SRB1 and NPCIL1 transporter channel, which that carotene will move inside, and in the presence of BCO1, which that is a retinaldehyde formation will be occur in the enterocyte. So this is the enterocyte of the duodenum. And here is the retinol. This retinol contain hydroxyl group will also enter through a retinol binding protein receptor 2, possibly through transporter channel, while also the retinyl ester formation also occur. And in this way, this was the gut, and after the entry into the enterocytes, carotenoids will can convert into the amphiphilic chylomicron. This chylomicron will enter into the lymphatic duct. So this is the lymphatic duct, and here is the portal vein. Portal vein means the vein will move toward the liver. So the liver contains double circulation. So here is the lymphatic duct, and in this way here is the retinol containing hydroxyl group will move into the portal vein and ultimately go into the liver. While here the chylomicron will move into the lymphatic system and also enter into the blood circulatory system. So this is the hepatocyte, suppose, in the form of liver, are present in the liver. And endocytosis through scavenger receptor bind after the remnant chylomicron, after the using into the tissue. So this remnant will move in endocytosis, is known as endocytosis, to become an endosome, and that will be retinol bind with the retinol binding protein 1, and after this retinol binding protein 1 will bind, and in this way the retinol again binding with the retinol binding protein 4 and TTR, and in this way it will be move and dissociate again with the retinol binding protein 4 with the TTR, and ROH will be move into the this binding protein will lead to movement into the tissue, different tissue. So that is why the retinol binding protein 4 is important. After the entry through a STRA, retinol uh, uh, A6, which that is basically is the Jack and Stat phosphorylation to become a dephosphorylation and again phosphorylation. So that is the mechanism, and this is the function for the reactive nitrogen species and reactive oxygen species inhibition and clearance. On the other hand, the lipid per oxidation inhibition. While the chylomicron will enter into the blood circulatory system, also go into the tissue with the retinol, with the with the carotene binding protein 1. So on the other hand, here is the chylomicron will transport the lipase. So the lipase enzyme used to different tissue will provide the lipids. And here is the eyes. Eyes is very important for the retina. So the retina is very important, contain rod and cone cell. Rod and cone cell contain rhodopsin, and that's rhodopsin is attached to the retinol, and this retinol is basically used for the absorption of light, to detection and accommodation of light and detection of the object through light reflection. So this is the retinaldehyde will be moved through a retinol convert into the retinaldehyde, and this is the cytochrome P26A1, which that is used for the conversion. So in this way, here is also the hepatic stellate cell, which that is also used for the reserve of the retinol with the carotene binding protein 1, and this is the retinal ester formation will become, and this is the basically the loading of the retinal, and here is the LRAT is used this enzyme for conversion of the retinol into the retinal ester. So this is the walds visual cycle is very important for the transportation. This is the rod cell, and transportation the vitamin A into the rod cells of the retina in the eyes. So this is when light will be produced. So the rhodopsin is important for give the nerve impulse. So remember about that. After this, the how to become a rhodopsin. So first of all, it will be give the nerve impulse and separate two molecules after the dissociation and inactivation. So this rhodopsin is the 11 cis retinol opsin contain basically the retinal opsin. So the retinal contain opsin, which that become a rhodopsin, which that is again all trans retinal dissociation to become a opsin again formation and all trans retinal dissociation, and alcohol dehydrogenase enzyme used to NADH in the liver. NADH used to reduce into the NAD oxidized, so this is the alcohol dehydrogenase enzyme used to produce the all trans retinol. So the alcohol dehydrogenase enzyme used to produce the all trans retinol because the hydrogen addition. So the isomerase enzyme also present in the liver, which that will convert the 11 cis retinol. This 11 cis retinol again move into the rod cell, and move into the rod cell to 11 cis retinal conversion through a NAD plus to convert a NADH. So the oxidation. So in this way, this mechanism is known as the Wald's visual cycle, which that is used for the vitamin A through a opsin, and the opsin combined with the retinal to give a rhodopsin, which that is important for the light sensitivity. So here's the vitamin A. This vitamin A basically is the antioxidant and the free radical scavenging, reactive oxygen species and reactive nitrogen species inhibition, and prevent the lipid peroxidation. And the beta carotene is the important vitamin A, which that inhibit the sodium and potassium stimulate and catalase, and basically is the GS transferase enzyme. Basically is the present, and the fucoxanthin break free radical chains and stimulate the glutathione. So this all thing is about the vitamin physiology. Next video we will discuss further. So thanks.